So students, we will be discussing about the eukaryotic cells. Eukaryotic cells include all the protists, including unicellular eukaryotes, plants, animals, and fungi. It has an organized nucleus and it contains hereditary material which is covered on the outside by the nuclear envelope, which is a double membrane cover. Now, all eukaryotic cells are not identical. The plants and animal cells shows differences. Let us discuss only about the different components of the eukaryotic cell in this chapter. So, we will start with the outermost covering, that is the plasma membrane. The plasma membrane is the outermost covering for the animals, and for the plant cell they will have an additional cell wall around them. So, when we just talk about the plasma membrane, the most improved model of the structure of cell membrane was proposed by Singer and Nicholson. These were the scientists in 1972. And this model, according to this model, the fluidity of the plasma membrane is due to phospholipid bilayer. So it has components called phospholipid, which is present in two layers. So phospholipid bilayers, which is rich in unsaturated fatty acid, and it forms the main component of the cell membrane because it provides the structural framework to the membrane. So these lipids, they are amphipathic. Amphipathic means that they are structurally asymmetrical with a polar hydrophilic head and non-polar hydrophobic tail. So this is the hydrophilic end, and this is the hydrophobic end or tail. And on the outer side of some of the lipid, sugar chains can be also attached. So in them certain sugar chains can be attached, which I can show you. So certain sugar chains may be attached like this, and these are glycoproteins and glycolipids. So some can be glycolipids and sometimes glycoproteins. There are two kinds of proteins that are found in this membrane. So let me first complete drawing the bilayer. So this is the phospholipid bilayer. This is one molecule of phospholipid, and like this layers of phospholipid will be present in two different layers. Apart from this phospholipid bilayer, there are certain proteins. So some proteins, they are present only on the periphery of this membrane, and they are called as peripheral protein. Peripheral proteins. So let me complete drawing this membrane. Now there are also certain proteins which will integrate deeply inside this membrane. So these protein which is transversing the both the layers of the phospholipid, it is called as integral protein. So there are two kinds of proteins, peripheral protein and integral protein in this lipid bilayers. And this model is essentially the fluid mosaic model. So students, I hope you have understood. So our next topic would be cell wall. The cell wall, it was first discovered by Robert Hooke. While observing the cell walls in cork tissue, and it is the outermost rigid, protective, non-living and supportive layer found in all the plant cells, bacteria, cyanobacteria, and some protists. It is not found in animal cells. The cell wall is made up of various components. For example, in algae, it will be made up of cellulose, galactans, mannans, and certain minerals like calcium. Calcium carbonate is a very common mineral present here. Then in case of fungi, the cell wall has a component called chitin. Then in case of the prokaryote, we know that the cell wall is made up of peptidoglycan. Then in between two plant cells, suppose this is one cell and this is the other cell. So in between two cells, cell 1 and cell 2, there is first a middle lamella. This middle lamella is the first layer which is present, and on both sides of this middle lamella there will be deposition which are formed. These depositions will form the primary cell wall. The middle lamella will be formed by the deposition of calcium pactate from both the sides of the cell, and then the cell wall will be primary cell wall will be deposited in the form of cellulose. So this cell wall will be formed around the plant cell. Also, apart from this primary cell wall, this is the primary cell wall, there can be also deposition of secondary cell wall from inside the cell. There can be many layers of secondary cell wall which can be formed inside the primary cell wall just by deposition of chemicals from inside the cell. So this is the secondary. There can be certain pits, or there can be certain connection between both the cells, cell 1 and cell 2, in the form of plasmodesmata. So in between two cells, sometimes we see that the plasma membrane will remain fused. The plasma membrane of two cells remain fused, and this is called as plasmodesmata, which forms a tunnel-like structure for transporting materials between the two cells. So this is called as plasmodesmata. So it is the connection between the two cells. So we know that the cell wall may not be completely sealed from all sides. There can be in some cells there will be pits. For example, in tracheids or vessels in xylem, as we know, they will have pits. Then in the sieve tube, we know they will have also certain pits and holes present on the cell. And in many of the living cells, they will have plasmodesmata in between them. This is also called, there can be also cytoplasmic strands. Then the function of this cell wall will be to provide protection and to provide a proper shape to the cell. Also, apart from that, transporting necessary substances into and outside the cell can take place through this.